that is because one of our points in the syllabus, so the way we're doing grading is we have a bunch of different categories. So you have your individual online quizzes, the discussion posts that you're doing on your own. Then you have the team online quizzes, your exams, and these team assignments that we're doing. At the very end, there'll be a team evaluation. We'll talk more about that in a little bit. But this is just basically your teammates will score you, like, did you show up? You know, did you do some work? Things like that. But what we're also doing is your lowest score is going to get dropped in the individual quizzes, in the team quizzes, and in the team assignments. So if it looked like you completed something, but the score wasn't being calculated in, that's because we did one team quiz and one group quiz. So they wouldn't have been factored in yet because they were all your lowest. So after today, that'll look more normal. Okay. Other questions about this grading scheme? I'm not a big fan of reading the syllabus out loud to everybody, but I realized that I maybe skimmed a little more than I should in lieu of making sure everybody understood how the Brightspace course worked.
Is that just why it says dropped next to like our first quiz right now? Okay.
Yeah, it's because that's your only quiz, so it's your lowest quiz. Yep. Okay, sounds like we're okay on that. Okay, so I'm going to start us off by talking about our first two macromolecules so that you have the information you will need to complete both of the problem sets we're going to be working on today. So let's get into it. So one of the main reasons we cared about doing a review of chemistry at all is that what we really care about is how biological macromolecules, so like the organic molecules that make up cells, are going to behave. And we're going to see that we have 4 types. So we're going to have carbohydrates, lipids, proteins, and nucleic acids. We're going to go over 2 of them today and 2 of them on Wednesday. So you won't have to turn in your total biological macromolecules problem set until Wednesday, because we're going to do half of it now and half of it later. All right. So we're starting with our 2 groups, our carbs and our lipids. So these are the first 2 on our list: carbs, lipids, proteins, nucleic acids. All of these different classes of macromolecules are going to be considered organic molecules because they all contain carbon. They'll also contain hydrogen, oxygen, potentially nitrogen, and phosphorus, depending on specifically which one we're talking about. Backbone, super important that it's carbon. We want to remember carbon is always going to form 4 bonds, 4 covalent bonds. Okay, so it's going to be the backbone for all of these different types of molecules. Macromolecules also are different sizes. We're going to have lots of different types of carbohydrates, lots of different types of lipids, lots of different types of proteins, but they're all going to be made up of smaller parts that we can recognize. So the individual subunits of a macromolecule are not just the atoms in there, there are monomers, these little building blocks that we're going to string together into larger molecules we're going to call polymers. So in this case, what we're looking at is building a carbohydrate from 2 monomers of carbohydrates into a larger carbohydrate, in this case, a disaccharide, but a big polymer of a carbohydrate would just be a lot longer, basically. So we can see what's happening here. When we link monomers together, we link them together by forming a covalent bond. So here, highlighted in red, we can see that the different parts of these monomers that end up joining together for our covalent bond. So we can see that we took this OH group and this hydrogen. We pull them off. They come off as water, leaving behind a covalent bond from one corner of this molecule to this oxygen. And then we have this remaining covalent bond back to this ring structure. So we formed a covalent bond, a new covalent bond here. We lost an oxygen and a hydrogen from here. We lost a hydrogen from here, but kept that oxygen there. Okay. So specific to monomers always build into polymers like this by forming covalent bonds between their subunits. The specific form that we see linking here, this covalent bond out to an oxygen and then a covalent bond into this ring structure, is very characteristic of carbohydrates. So if you ever see a picture of this, I like to think of it as like kind of rings holding hands with an oxygen in the middle, like your fist is kind of the oxygen. That's going to tell you you're looking at a carbohydrate. Just a fact to know. We'll get into that in a second. This is a table listing the names of monomers and polymers, like what we would call a monomer of a carbohydrate versus a polymer of a carbohydrate, and the same for each of these groups. So our monomers for carbohydrates are going to be monosaccharides, mono for one. Polymers are going to be polysaccharides, poly for many. Saccharide means like sugar. Lipids are weird, so we're really going to mostly refer to our lipids as hydrocarbons. We'll see that that's because they're made of hydrogen and carbon, almost exclusively. Proteins will have amino acids, polypeptides. Nucleic acids will see nucleotides, and then DNA and RNA. We'll talk about those on Wednesday. I just like to have it all condensed. Okay, so starting out with our carbohydrates, each of our macromolecules play a different role in the body. Our carbohydrates their main purpose is to provide energy. Specifically, they're providing energy in the form of glucose, which is one of our monosaccharides, so one of our monomers. And glucose is found in grains, fruits, and vegetables. There are other types of carbohydrates which are processed in similar ways, but for this course, we're really going to be focused on glucose, and honestly in your future courses as well. We'll talk a lot more about glucose when we get to metabolism at the end of the course. All of our carbohydrates, however, are going to be represented by the general formula CH2O, all in parentheses, N. What that's telling you is the ratio of atoms in our carbohydrates. Oops. Okay, so it's telling you that we have this 1 to 2 to 1 ratio of carbohydrates. Lots of hydrogen, but we also have equivalent amounts of carbon and oxygen. Right? So our monomers are the monosaccharides, our polymers are the polysaccharides. We're going to look at some examples of what our carbohydrates might look like because we want to be able to identify these macromolecules when we see them. And that's going to be a large part of the goal of our problem set, is looking at example macromolecules and telling which of these 4 groups they're part of. Okay. So monosaccharides are monomers of the carbohydrates, can be found as a linear chain or as ring-shaped molecules. They form rings in aqueous solutions, aka solutions that form water, or that are in water. So basically in our cells, they're always going to be rings because the fluid surrounding our cells is water and the fluid inside our cells is water. So the most important for our purpose is these ring structures. So this shows you how a linear carbohydrate would kind of curl up and form a ring. That's not that important to understand for right now, so I'm going to kind of gloss over it. But what I do want to make sure that we all remember is when we're drawing out molecules, if you're pretty far into chemistry thus far, you should recognize this, but whenever we see an angle, like a point, and there's no atom drawn at that angle, that means there's a carbon there. It's just a shorthand. And we'll see a lot when we move to lipids. If you can't count 4 covalent bonds coming off that carbon, you should just assume that there are hydrogens attached to it, adding up to 4 total bonds off the carbon. There are various like abbreviations like this we use when we're drawing molecules, especially in biology, because these molecules get big. Okay, but we're looking for rings when we're looking at carbohydrates. We expect to see plenty of carbon, plenty of hydrogen, and critically, plenty of oxygen. So the presence of this much oxygen is something that should make you immediately suspicious that you are looking at a carbohydrate, as that's going to distinguish it from a lot of our other macromolecules. We also want to talk about functional groups that we'll see in our different types of macromolecules. That's because these different functional groups have different properties. So we talked, when we were talking about chemistry, about electronegativity and how certain atoms kind of hog electrons even when they're in a covalent bond. So oxygen in particular forms polar covalent bonds. It hogs those electrons, so the oxygen is slightly negative. So we'll see that the presence of all this oxygen in carbohydrates makes them act as polar molecules because they have all these polar covalent bonds, which basically means for us that carbohydrates are going to be kind of attracted to water. They're going to act almost similarly to the way our ions will act, which are our charged particles that we get when ionic bonds dissolve in water. And we'll talk about why that's important as we start to talk about transport and as we talk about cell membranes. So here we can see a polar carbonyl group, right? Polar because of this oxygen. And we can also see a hydroxyl group, right? So OH, this oxygen, is going to make those bonds polar, right? So we can see those there. Onto our bigger carbohydrates. So our polysaccharides are going to consist of a bunch of monosaccharides, so a bunch of our monomers linked together. And they can be any different type of monosaccharide, right? It's not always going to be a chain of just glucose. We have a couple of different types of monomers we can build together. Doesn't matter. If we have a chain of monosaccharides, it's a polysaccharide. A thing we'll notice when we're looking at our polysaccharides is that sometimes we're going to have just a linear chain, right? So just ring next, holding hands with another ring, holding hands with another ring, all in a long unbroken chain. Sometimes we're going to see branches off of those. So we don't always have to form that covalent bond just at these sides. We could make a branch go up or down, form a covalent bond, and this would still be a polysaccharide. So we'd call those branched polysaccharides. The other form where we just have a straight line of monosaccharides linked together would be called an unbranched polysaccharide. So we see some functional differences in these as well. So some are going to be storage polysaccharides, which are going to be adapted basically for storing sugars for use later. And this has to do with the use of carbohydrates as energy. So we'll see that amylose and a molecule called glycogen store our sugars together, right? Glycogen specifically stores glucose subunits together in human bodies, and these will later be able to be released as energy. Amylose is really starch, so starchy foods. That's our plant storage molecule, which again then can be broken up for energy, right? That's why starches are a good quick form of energy. Some structural polysaccharides do exist as well. Cellulose is a carbohydrate. It's found in plant cell walls. Okay. So structural meaning like it would give that cell wall structure, it would make it hard, make it stiff. Okay. So our next group will be the lipids. So lipids are kind of weird. Lipids is a group that involves a bunch of different molecules that look pretty different if you draw them out on a sheet of paper. But the thing that's going to unite them is that they are non-polar, so they're hydrophobic, they repel water. And they're non-polar because they are hydrocarbons. When I say they are hydrocarbons, what I mean is that they're almost entirely made of just carbon and hydrogen. If you flip back to that electronegativity table at the end of our chemistry PowerPoint, and see that hydrogen and carbon have pretty similar electronegativity. So they share their electrons pretty equally, meaning that there are no little charges building up anywhere. And so there are no little charges for water to interact with. So that's where we get this nonpolar property from. It's because our water is kind of charged on opposite ends. Our lipids don't have charges collecting anywhere, so there's nothing that really wants to interact with the water. This means that our lipids are going to reflect that in the ways plants, our bodies, whatever, use them. So they'll be creating sort of water repelling coating on the outside of animals. We'll see that if they mix into your blood or something, they're going to form conglomerates, which we'll have to break apart in specific ways. They're always going to be repelling water, at least in some ways. We'll get into a special lipid called a phospholipid that has a nonpolar and a polar part, which is going to be a critical part of our cell membranes. Okay. So lipid functions include providing insulation. So like fats can provide insulation as well as providing long-term energy stores. Our lipids are also going to form the basis of cell membranes. Some of them are going to be the building blocks for hormones. Hormones are actually come in two types, lipid and protein. So protein we'll talk about later. Lipids are going to be hormones as well. And they'll protect organisms from water, from wind, from temperature. We have 4 kind of basic types of lipids. So if we were making a chart of macromolecules, right, we had our 4 types. Lipids would have 4 branches. We got our fats and oils, we got our phospholipids, we got our steroids, and we got our waxes. And these pair pretty well to these functions. So fats and oils provide insulation and long-term energy stores. Phospholipids, basis of cell membranes. Steroids, building blocks for hormones. Waxes, protection from water, wind, and temperature. Although we can see mixed function depending on what we're talking about. So the most characteristic, the most common type of lipid that we see drawn out are fats and oils, and specifically are triglycerides. So triglyceride is a lipid made up of a glycerol backbone. So glycerol, if it weren't attached to all these chains, we would actually see as a carbohydrate. But when glycerol is attached to 3 fatty acids, that's where we get the tri from. The tri is for our 3 fatty acids. The glyceride is from the glycerol. Those fatty acid chains, these chains coming off, I think of this as kind of a flag, like 3 chains, are made pretty much entirely, right? Carbon bonded to a carbon with 2 hydrogens coming off it, bonded to another carbon, more hydrogen, more carbon, more hydrogen, and so on. So that's what I mean when I say that our lipids are hydrocarbons. They're almost entirely carbon and hydrogen. You'll note that other than this backbone, there's no oxygen anywhere in this. That's what makes this a lipid. We got these carbons and hydrogens. They have non-polar covalent bonds between them. So there are no charges, not attracted to water. They're going to repel water. There are different types of fatty acids. So that triglyceride just has 3 fatty acids coming off it, but sometimes we're going to see slightly different versions of what that chain looks like. Our first is a saturated fat. So a saturated fat just has single bonds between all our carbons. So every carbon has to make 4 bonds. So if we take some example carbon, we'll see it attached to a carbon on the left, a carbon on the right, and then its remaining 2 bonds each hold a hydrogen. And we call this saturated because this is the maximum number of hydrogens you could fit into that chain, right? Because we have to form the backbone with these carbons. There would be no way to get like 3 or 4 hydrogens on here and keep that backbone. But we do have fatty acids which will have fewer hydrogens. Those are unsaturated fats. So an unsaturated fat is going to have a double bond, right? So let's take this left one. Still has a carbon to its left, still has a carbon to its right, forming this backbone. But this bond on the right is a double bond to another carbon, meaning it only has room for 1 hydrogen left on it. So that's less than the maximum number of hydrogens this fatty acid could have, making it unsaturated. We'll note that this creates a bend in the chain due to the way the hydrogens are positioned. So we'll also see something called trans fats exist. So when we form this double bond in nature, we usually form cis fats, where our hydrogens are on the same side. In a trans fat, those hydrogens, we still don't have the maximum number, so it's still unsaturated. But those hydrogens are on opposite sides, so we don't see this bent structure to the fatty acid. And these are usually produced industrially and have health concerns because they kind of mimic this structure of the saturated fatty acid, this straight line. So our fats and oils are insoluble in water. We kind of touched on why this is, right? So we're looking at the fact that we have all these carbon-carbon bonds. The other bond we really have is carbon-hydrogen. That's it. So that's a non-polar bond, which means the molecule overall is non-polar, which means it's going to repel water. Water can't get in there and mix it up with the fatty acids. This changes a little bit when we look at our next group of lipids, which are our phospholipids. So the phospholipids are going to include a phosphate group. So we can see our phosphate group here. It's a bunch of oxygen. It's got an R group. An R group is just a variable group. It means we don't know what's here. And they have a phosphate in the center, a phosphorus in the center. So these phosphate groups are actually going to be charged. So it's even like more powerful than just a polar molecule. It has an actual charge because it ionizes to release hydrogen. We don't have to worry about that part. Okay. So when we look at a phospholipid, we're going to see a familiar structure at the bottom. We're going to see those fatty acids that we were just talking about. So this is part of why I was telling you that every time you see a point, you should expect a carbon. And every time you don't see a carbon with 4 bonds, you should assume there are more hydrogens attached to it that they didn't bother to draw. That's why this zigzag line tells you at every zigzag, there's a carbon at that corner with 2 hydrogens coming off of it because we didn't draw it in. Okay. So that part is familiar, right? This is our fatty acid chain, just like in our triglyceride. So we know those bonds are nonpolar. It's going to be hydrophobic. The other end of our phospholipid is made up of this phosphate group attached to those fatty acids with a glycerol, just like in our triglycerides, right? So our phosphate groups are charged. So this part of the phospholipid, we're going to call the hydrophilic head. It's going to be hydrophilic basically because of this charge and because it will often have polar groups attached here at the R group. This means that we have a new vocabulary term for our phospholipids. We refer to them as amphipathic. And what that means is that they have a part that's nonpolar and they have a part that's polar, basically. So they have a part that's attracted to water, they have a part that's repelled by water. Have a part, yeah, so they're going to have different ends that interact with water in different ways. That's because they have these 2 fatty acids that are nonpolar, don't want to be near water. They have this phosphate group that is attracted to water. So these are just some more facts. I said these things. Cool thing about phospholipids, and super useful thing for biology and life as a whole, is that because we have these different ends to our phospholipid, because they're amphipathic, because they have a hydrophobic end and a hydrophilic end, that means they're going to orient in very specific ways if we took like a bucket of phospholipids and just dropped it in some water. One of the main structures they're going to spontaneously form is something called a bilayer. And this is going to be the basis for our cell membranes. If we thought about it, we could predict that phospholipids would form this structure just by knowing that single fact about phospholipids, that they have a hydrophobic end and a hydrophilic end. Because what a bilayer is that we have these polar heads, these phosphate groups, right, on one side of our phospholipid. Here they're in the beige in this figure of this membrane bilayer, or here we can see them in red on this more chemistry style drawing, right? So those polar heads, they're attracted to water. So they're going to face any water we have in our beaker or in our body, right? If we just had one layer though, right, we just dropped phospholipids in and say they formed a single layer, right, that would be fine on one side of this phospholipid layer, right, those polar heads touching water. On the other side, we still have more of our beaker of water, right? But we have these nonpolar tails. They're hydrophobic. They really do not want to be anywhere near that water, which means they can't be exposed, right? They are not going to be happy if they're just sticking out and there's water touching them. So that's why we have a bilayer, bi for two, because we take the single layer of phospholipids in order to protect those hydrophobic nonpolar fatty acid tails. You just form another layer directly facing it, right? Then those nonpolar fatty acid tails, they're not touching water anymore. They're just touching more nonpolar fatty acid tails. And they're protected by this second phosphate group on the opposite side that can then touch the rest of the water in the environment. This is going to be really useful in a cell because we have water inside the cell and we have water outside the cell. So both of those water-filled environments are going to be contacting a phospholipid polar head. We'll talk more about phospholipids in detail when we talk about those cell membranes. So our third type of lipid is a steroid. So steroids are based on this specific steroid called cholesterol, which we'll see in our cell membrane. And all our other types of steroids, our named steroids, are basically going to be modifications of cholesterol. So whenever we look at a steroid, we're going to be seeing 4 rings linked together with some additional groups coming up here off the side. Now, this ring structure can be a little tricky because sometimes we'll look at this immediately and we'll be like, oh, I see rings, it's a carbohydrate. I know carbohydrate has rings. That's a good first guess, but we want to be particular about this and notice that those rings, they're not holding out their hands with an oxygen in the middle. They don't have this sort of spaced set of covalent bonds. Instead, these rings are directly pressed against each other. We can actually see that those rings would be connected in multiple places. And there will always be 4 rings. It's always 4 rings pressed together like this if we're looking at a steroid. And then we see the hydrocarbon part coming off the side, right? So this is 3 hydrogens and a carbon. Then we just got more carbons with hydrogen sticking off the side over here, right? So we have a lot of hydrogen and carbon. All we really see on here is 1 oxygen, right? That's not enough oxygen for a carbohydrate. So we want to be careful when we're looking at these molecules to figure out what's going on with oxygen. Do we see any? Do we see enough? So cholesterol is going to be a component of cell membrane, so it's the steroid we'll talk about most often. But some hormones are also going to be steroids. Estrogen and testosterone, the sex hormones, are some really important steroids in terms of body function. There are going to be slight differences in how steroids behave depending on the chemical structure. So by that I mean depending on exactly what is attached here, kind of off to the upper right of those 4 rings. So we will see slight differences in how they behave depending on what's there. But for the purposes of just learning our macromolecules, we don't need to know about that. Okay. Our final lipid type is waxes. So waxes are just long fatty acid chains that kind of bent in the middle and then swirl backwards. They're different from flats because they don't have a glycerol backbone. So when we looked at the triglycerides, right, it had that kind of flag structure, the glycerol backbone, and then 3 fatty acids coming off it. A wax is really just going to be the chain Maybe there'll be something in the shape, but that's it. Very simple. Those are going to be coating things like the surface of plant leaves to prevent too much water from escaping, things like that. Okay, so I am going to click out of this here. The remaining slides are kind of some instructions for helping you with the problem set, but I am going to open up the problem set and walk through that real quick to make sure we all know what we're doing. Okay. So this is not due today because we're going to talk about our additional macromolecule types as well. So we have two basic things we're doing. So one is that we're going to be creating some concept maps. So like you're going to talk about carbohydrates. You're going to take some of these terms that we just learned and you're going to link them together. Now, the video has instructions about concept maps, but basically a good concept map is going to have arrows between the terms that make sense, that you could basically put a verb or an explanation on versus just kind of loose associations. So as you are making your carbohydrate concept map, you want to think about like why you're linking together two things, why you're putting branching points there. Okay. So we're going to make a concept map for carbohydrates and we're going to make one for lipids. The other thing we're doing, and you can start here if you want, doing this in a specific order is not that important. We're going to be looking at these examples of macromolecules. So here we see an example of a glucose molecule and kind of like a fact sheet sometimes about them. So here we see hemoglobin, we see what it looks like, we see some facts, insulin, and so on. So each of these pages is an example of a molecule with some information about it. And then when you come back to this, you're going to be filling out this chart. You're going to say what type of macromolecule it is, and you're going to say why you think it's that type of macromolecule. So there's not necessarily a specific correct answer for this evidence column. It's just like, how did you tell? Like, why did you do this? And you should not need to Google this. I know I did say that Google is a fair source for problem sets in general, but for your own learning, you should be able to figure this out just from the information provided. These last couple ones do kind of trip people up sometimes. So I'm just going to tell you right now that you can have more than one of these macromolecules in some things. Okay. So that should get you started on here. So we're identifying these molecules. And for right now, if you look at something and you're like, that's not a carb or a lipid, skip it and move on to the next one because we'll be doing proteins and nucleic acids and filling those in here later. Okay. So that's what's up with the macromolecules problem set. Quick thing, just back to the chemistry, because you should be working on that. If you didn't finish it, please turn it in by the end of today. One question I got from a bunch of groups was about what you should be doing when I say to draw the molecule in water. What we're getting back to there is when we're thinking about ionic compounds, we're thinking about the fact that they dissolve in water, they dissociate into these structures called ions, and then water molecules orient to them in very specific ways because of the polar bonds in the water molecules. So you want to draw something like this for your ionic compound, paying close attention to how the water is interacting with the ions you formed. Okay. So I'm going to send us out into our breakout rooms now and call me over if you have a question. Otherwise, I'll kind of pop around if I don't hear anybody begging for help.